Hello everyone, welcome to BS 3336 UAB Proteomics Workshop on the topic of Glycomics and Glycoproteomics. In this video, we will be talking about glycosylation and how it affects the structure and function of the protein. And next, we will be talking about what is Glycomics and Glycoproteomics and how to make use of the proteomics analysis to analyze and detect the protein modification through MS spectrum and database search. And lastly, we will be focusing on the case study about how glycan can be used as a diagnostic biomarker to detect particular disease.
Glycosylation process is a covalent attachment of oligosaccharide chain on the protein backbone. It is considered as the most common post-translational modifications of protein. There are two different ways in which glycan can be added into amino acid. Through enzymatic ways called glycosylation, where this process is done by enzyme called glycosyl transferase, and the other one by non-enzymatic ways in the process called glycation. This diagram illustrates the cellular processes of protein glycosylation. Most plasma membrane and secretory protein contain one or more carbohydrate chain synthesized through the glycosylation. In cell, some glycosylation reactions occur in the lumen of the endoplasmic reticulum, while others are in the lumina of Golgi cisternae. Glycosylation can be categorized under different types: N-link, O-link, glycation, C-link, and phosphoglycosylation. In N-link glycosylation, the glycan binds to the amino group of asparagine in the endoplasmic reticulum. In O-link, it is characterized by monosaccharide which bind to the hydroxyl group of serine and threonine in the ER, Golgi, cytosol, and nucleus. In C-link glycosylation, the mannose binds to the indole ring of tryptophan. And lastly, in phosphoglycosylation, the glycan binds to serine via phosphodiester bond. In summary, the glycosylation categorization relies on the signature amino acid residue it binds to. Now let's go deeper beyond the categorization. From this figure alone, you could have probably imagined that there are many different ways on how each monosaccharide can be arranged within a single glycosylated amino acid. You are right, there is no tell on how many different combinations of different glycan can be added. This is achieved just by simply adding different monosaccharides on top of the constant base pattern. For example, in N-link glycosylation, the chain in closest proximity with the amino acid is always a linear 2N-acetylglucosamine followed by a mannose, and then split with 2 mannose attached to each branch.
Glycosylation causes the post-translational modification through the addition of carbohydrates on the protein structure. Glycomics is the comprehensive study of glycomes, which involve the entire carbohydrates in the cell. Glycomics is the analog to genomics and proteomics, which is the systematic study of all glycan structure of a given cell type or organism. The glycome exceeds the complexity of the proteome as a result of the even greater diversity of the glycomes constitute carbohydrates, and it's widely accepted that protein glycosylation is involved in numerous essential cellular processes. Thus, the structural and functional characterization of these post-translational modifications are important in the rapid growing research area of glycomics and glycoproteomics. Glycoproteomics is the study of glycome attached to the proteins. It is the branch of proteomics that identifies, catalogs, and characterizes proteins containing carbohydrates as a post-translational modification. Glycoproteins also study the change or modification of protein which is associated with the disease using the combination of conventional glycan analysis through MALDI-TOF and glycopeptide analysis through LC MALDI-TOF. This is another example showing MALDI MS spectrum of N-glycopeptide detected from bovine IgG. From the same MS spectrum, detailed information can be derived on the peptide mass, peptide sequence, and the glycan structure respectively. As discussed earlier on, glycosylation is the most common post-translational modification of protein. plays an important role in protein folding, interaction, stability, as well as signal transduction. By regulating protein activity, glycosylation is involved in the normal functioning of the cell and in development of diseases.
This figure depicts the experimental workflow of typical glycoproteomics study in laboratory. It is a top-down approach to study glycosylation from its interconformation with peptide or glycopeptide, all the way down to the saccharide composition of the glycan itself. The process starts with sample preparation, where complex sample mixture has to be purified to extract the sample protein. This step is important to reduce the noise in subsequent analysis. Trypsin digestion is utilized to separate the glycopeptides from non-glycopeptides for the interest of this study. And then, the peptide sample will be further processed by peptide and glycosidase F, also known as PNGS-F digestion, which is specifically useful to separate aniline glycans from other type of glycans. And finally, all of these processes will be analyzed using multi-top MS-MS or LC-MS technology for glycan localization within peptide and its saccharide structural composition. The information about glycan structure can be derived by two MS approaches called top-down and bottom-up. In a top-down approach, an intake glycoprotein is analyzed directly using electron transfer dissociation (ETD) or collision-induced dissociation (CID) mass spectrometry technique. ETD induces backbone fragmentation, resulting in a mass spectrum that reveals the information about peptide sequence and glycosylation sites. On the other hand, CID favors the fragmentation of glycosidic bonds, providing information about glycan composition and the branching. In a bottom-up approach, the glycoprotein is first digested by an enzyme called pronase E to yield the glycan moiety attached to a single amino acid. This glycan product is then analyzed by NMR to acquire structural information about the glycan, or by MS and MS-MS to acquire sequence information.
After treating the sample through MS analysis, the spectra could be further analyzed in conjunction through glycoinformatics. Due to the advance in experimental analytical methods such as mass spectrometry, there is a tremendous increase in the amount of carbohydrate structure data generated. The availability of databases and tools to store, retrieve, and analyze these data in an efficient way is of fundamental importance to progress in glycomics. For glycoprotein and glycopeptide sequence analysis, a large number of well-characterized and annotated glycoprotein are commonly found in the UniProt knowledge base. Many glycopeptide mass spectra also expanding the Peptide Atlas library, which stores millions of high-resolution peptide fragment ion spectra acquired from a variety of biological clinical samples for peptide and protein identification. Ultimately, the data obtained from different aspects of the workflow need to be merged and interpreted in an integrated fashion so that the full extent of glycosylation changes associated with a particular biological state can be better reviewed. However, GlycomDB is an integration of many other databases that is essential in studying glycan. This would provide a holistic view of the query search. A library has been implemented for the interpretation of different encoding schemes, carbohydrates. This diagram shows the typical workflow for identification of cancer biomarker in the patient's serum during diagnostic step. First, the serum is taken from patient or normal individuals, and then the sample is treated and sent for mass spectrometry analysis, which would then computed for the database comparisons, such as the detection of glycomic signature based on the expression level and structural changes. The study of glycoproteomics has provided a significant impact and also promising result in medical field, especially in the study of human diseases through the expression of glycans. Glycoproteomics is a state-of-art technology that are in current use to describe and quantify changes in glycome, as well as the application of this technology to the study of specific cell, tissue, and organism. With the development of technology and methodology, the details of post-translational modification in particular glycan structure can be identified, which accelerate the discovery of glycan function. By integrating the knowledge gained from glycoproteomics, it provides better understanding of glycan in physiology, development, and diseases, which would provide a breakthrough in disease treatment.